All right, so welcome everyone to this 23rd workshop of the Agricultural Genome to Phenome Initiative. I'm Nicole Scott, I'm the project manager for AG2PI. Today's workshop is CartograPlant, integrating, visualizing, and analyzing genotype, phenotype, and environmental data from geo-referenced plants. This is the result of an AG2PI seed grant to Dr. Irene Cobo and this team that you'll be meeting today. And AG2PI as a project is funded by USDA NIFA. As just a couple more people are joining, I just want to note that the team here has a great schedule for you with some Q&A built in. But if you do have questions, feel free to just pop that into the chat at any time, and they will address those in a timely manner. Just got more people coming in. So just to get started, Dr. Jill Wegerson will be starting off today's workshop, and she'll be introducing everyone as well as it's their turn to talk, because you get to hear from everyone today. So Jill, do you want to go ahead and take it away?
Absolutely. Thank you, Nicole, for the introduction. I'm going to go ahead and share my screen here. And everybody can see that okay? Perfect. All right, so today we're going to be talking about CartograPlant, and I'm very excited to see so many people in the Zoom space with us today. We have a whole wonderful team that supports what we're going to be talking about today. So I actually wanted to start first by introducing that group. So you mentioned, Nicole mentioned Irene, who was a postdoc with us and now a postdoc in Madrid. And she's going to be talking a lot about the analytics part of CartograPlant today. Rich is our lead developer of the CartograPlant application. So he's going to be giving us a little bit of a behind the scenes view of what's going on. Vlad's not with us today, but he's one of our primary developers for our import pipeline. Emily is with us today. She's the lead database administrator for tree genes and also all the data in CartograPlant. Gabe is a newest member of our team. He's been focused on our analytic workflows. He's going to be talking to you today about basically the background of how all of this works for population genetics in CartograPlant. Sean is a triple developer. We'll be talking about kind of our relationship with triple. Shay is our coordinator, kind of our project manager for CartograPlant. Meg is with us today to talk about TreeSnap, and she's going to be talking about a very cool mobile application and how that data gets into CartograPlant. And then I want to highlight our biocuration team. These are undergraduates and also recent postdocs that do a lot of the heavy lifting in terms of how the data gets into CartograPlant. But we're going to try to make their lives a lot easier, and we're going to talk to you guys about how to do that as well. But a huge shout out to them and all the hard work they put into this. And I also want to acknowledge Nick Herndon. He's a postdoc, a previous developer on this, and now a professor at Eastern Carolina University. So that's a whole lot of awesome people doing a ton of work. So we're really excited to share all of this with you today. So our schedule, and I should say this is an Eastern time today, and I know we have people joining from all over the world. So good morning, afternoon, and evening to everybody who's here. As I said, I'm going to start us off today with sort of an introduction into CartograPlant. Then we're going to hear from Emily on how we get data in and what is in CartograPlant right now. Meg is going to take over from there and talk about TreeSnap in terms of data collection and mobile phenotyping. And then we're going to get into the weeds with CartograPlant a little bit more after the break, a little bit of the technical aspects with Rich. And then Gabe and Irene are going to talk about how to do some analysis in this application. So what is CartograPlant? So this is essentially a web-based and what I like to say, a map-based platform that facilitates meta-analysis across scales. So our goal is to move essentially from genes to environment. And I don't think I have to tell anybody who's joined this call today that the way that plants are responding, how they're growing, how they're performing, whether we're looking at productivity or conservation or restoration, is very much tied to the environment as well as their genotype, and also all the other omics that we're starting to look at, things like metabolomics, things like methylation. And so all of this is a part of our understanding of plants and their relationship with their environment. And so CartograPlant is attempting to make that a little bit easier by integrating data across scales. So who needs this data integration? We'd like to think just about everyone, but a lot of the applications that CartograPlant is currently serving is questions related to invasive pests and pathogens, questions related to plant breeding, questions related to assisted migration, invasive plants, overall examination of climate adaptation, and we also have, with a lot of our tree-related data, applications related to timber tracking as well. And so just to give a little bit of history on CartograPlant, I should say that CartograPlant used to be CartograTree. It's no easier to say now, but it is a little bit more comprehensive. And so it actually stemmed out of the Tree Genes database, which I always like to talk to USDA audiences and say this Tree Genes database, which was originally Dendrum, was one of the first 3 plant genetic databases that were funded by the USDA back in the early 90s. I said it was known as Dendrum then, but it is still in existence today. So it moved from kind of a piece of software that you could download locally and look at genetic maps. And now it is, of course, a web-based repository like many of our other crop species are as well. This database holds over 2,300 tree species today. And the reason I want to highlight this is because this database now, not when it started, but now is housed in the TRIPL framework. And so TRIPL is a software framework for biological databases. This framework composes a web front end, a database component that can interact with the Chato database, as well as an analytic component that is supported by the open source software Galaxy. And so TRIPL is actually supporting many different plant and animal and insect databases that are out there today. And it makes it very easy for researchers to basically install and get a database up and running, and also interact with other plant-based TRIPL databases. So there's a lot of instances of TRIPL databases today around the globe, at least 40 or 45 of them that I'm aware of. And here's just a view of some of those sites here. And so the reason I'm bringing up TRIPL in the context of CartograPlant is because this is the framework that our primary database is based on. And CartograPlant is a module that we've developed. One of the key aspects of the TRIPL framework is that each of these independent databases can contribute custom modules that are specific to their needs, right? So what I'm showing in green there, for example, is JBrowse. And so this would be a module that many users in the community would want, many TRIPL database instances would want, because it is a module to view genomes. But then in our case, we had some very specific needs of wanting to integrate genotype, phenotype, and environment data. And so CartograPlant was developed out of that, as available as a module. And also, there's an independent module that's responsible for getting the data into our database. And this is the PlantPopGen submit pipeline that we're going to talk about today as well. So that's just a little bit of the background of the framework that all of this is built on. And so here's a view of what CartograPlant looks like when you arrive. And the goal of CartograPlant is essentially to integrate genetic phenotypic data for geo-referenced plants through a map-based visualization environment. And we're integrating all of this with environmental data. And the secondary goal is to provide search capabilities as well as data analysis capabilities to all this diverse data so that you can actually ask biologically relevant questions in this framework. And so one of the strengths, I think, of CartograPlant is definitely in the environmental layers. And so we host a large number of global and regional layers that users can query and users can actually capture data from directly. You're going to be hearing a bit more about those layers from Rish today. And so when you go into CartograPlant, and you can head over there from cartograplant.org, and you go here, you can either, whoops, everything's moving fast. You can either get started and go into the CartograPlant interface, but you can also submit data. And we'll be talking a little bit about how to do that. And that's leveraging that other module, the Triple Plant PopGen Submit. And so here is a view of the homepage for, or once you go into the Get Started framework in CartograPlant. And Irene's going to be walking you through the interface even more, but just to give you a little bit of a preview, you have, when we're talking about the environmental layers, the ability to view those and turn on or off that panel. And then you can actually see a list. You can expand out that list to see what types of layers exist. And once you've selected a particular layer that will actually appear on the map, you can get information about where that layer came from, citation, description, what resolution is that layer available for. You can modify the opacity of that layer. And you can also see relevant legend information to let you know what a particular color or value relates to on that layer. Here's an example of canopy height layer. You can see that information viewed here. Here's a popular layer that a lot of our users will look at, which is the major soil groups. Here is the world CLIM layers, which are probably the most widely used layers for our analyses. And so here's an example of looking at precipitation and being able to view that on the map. What I wanted to point out here is that when you're kind of clicking around on the map, when you have any of these layers loaded, that you will get all of the values at that location for all of the layers that you have loaded at that time. In addition, you'll be able to have that information if you are looking at a particular plant that's highlighted at the same point. And so here's another view. Looking at that, let me just pull my screen over where we have the environmental layer loaded, and also we're able to click on a particular plant and view the detailed view for that plant as well. Another feature that we have within CartograPlant is range map information. And so at this time, I will say that a lot of our range map information is primarily focused on tree species, but this is something that we're looking to expand in the platform. This enables you to select if your species is available, load those range maps, and that can be very useful for a lot of different types of analyses that you may be doing. And so overall, the view for CartograPlant has a lot of layers. So we have over 900 layers available in CartograPlant right now. We kind of keep track of what people are using, and we also take requests for layers that people may want that we don't have available right now. So now let's talk about where the plants come from. So we've got a lot of layers. We have all this environmental data that's housed. You can search it, query it, load different layers in there. Now let's talk about the plants. So we kind of get information on these plants from primarily 3 different sources. The best one is number 1, directly from the scientists. And Emily will be telling you how to do all of that today. The second one is through mobile application. So this can be landowners, this can be citizen scientists, or this can be direct research. Meg will be talking to you today about that. And the third one is our curation. And this is actually where a lot of our data is coming in right now. And this is where our biocuration team does a lot of their tremendous work. And so what kind of studies does CartograPlant take in? We're interested in basically any combination of genotype, phenotype, data. It doesn't have to be genotype and phenotype. It can be one or the other with geo-referenced data. And when I say geo-referenced data, this can also be plots. These can be monitoring plots. These can be common gardens. And so we have the ability to take in a lot of different study designs. And so a lot of the types of studies that you might see would be things like population structure, landscape genomics, association mapping, diversity estimates, things like this. On the biocuration side, even though we have all of our journal articles in electronic form today, we no longer necessarily need to go to the library to find that particular journal article that you're looking for. From our perspective, this is kind of still what it feels like a lot of the time to try to bring this data into CartograPlant. And we work with repositories, which have been a game changer in terms of making this a bit easier. So things like Dryad hosts these flat file information for us to bring in, but we often are spending a lot of time trying to get those files into a format and understand what they are, especially kind of in the absence of having the original scientists there. So if you get a lot of emails from the biocuration team, they're just asking questions about how to get all of this information in. So just a reminder here within the triple framework, the pipeline that brings in the data, both from the public and also from the internal curation team, is through the Plant PopGen submit workflow. And so Emily's going to go into a lot more detail here, but this is just a view. This is a triple module. It has documentation in terms of how this data gets in. We're constantly updating how this pipeline works, so there'll be some new documentation coming soon. But the goal of this is essentially to make sure that we're asking all the relevant questions along the way and mapping all of that data onto ontologies and trying to make it as easy for the user as possible. So in the end, the goal is that we have the metadata, we have the raw data or links out to the raw data, and we have everything associated with a single long-term digital object identifier and meeting minimal reporting standards. So it's both a metadata and data intake workflow. And our primary goal is to make all of this data fair. So everybody is probably familiar at this point with this acronym: findable, accessible, interoperable, and reusable. And from this perspective, we're interested in providing persistent identifiers, using standard file formats, using metadata tagging, standard vocabularies, and also making sure that we're giving credit to everyone who's generated that data along the way and linking to all of the relevant aspects. And at a very, very high level, this is the type of information that we're walking a user through during that data collection process. So the first questions are things like, is it genotype, phenotype, or environment? What combination do you have? And by what I like to say is it's a choose your own adventure type of workflow. So in that way, you're not answering a lot of questions that are hopefully not relevant to your study. And by starting out with the high level aspects of the design, we're going to walk you through sort of the end questions that we need. And this was a lot of the area that we really focused on too as part of the AG2PI grant, was to really focus on refining and getting these questions right for the community. So you're going to hear more about that. And then I also wanted to just mention the reality of what it looks like. And I think Emily's got a couple other examples. So, when we're starting to look across all of these different studies that come in for a given species and figuring out what's mapped to the same genome, what's using a genotyping assay, and so on, this is the type of sheets our biocuration team is working with to try to figure out, are they using some of those phenotypes from that study? Are they generating new ones? And how do we put this all together? So this is where all the magic happens in terms of making this data fair. So as I mentioned, another way that we get awesome data in and phenotypic information in specific is through TreeSnap. So Meg's going to be talking about all of their work here. This is primarily focused on trees, but their team is also doing a lot of work and plans in the future to develop some applications that can expand beyond trees as well, that will also interact with CartograPlant. So that's kind of 2.0 plans as we move forward. Okay, so just to give you a view of when you're in CartograPlant, we talked about the ability to turn on and off those environmental layers. But I also wanted to highlight here that you have the ability to turn on and off these plant dataset sources that I just talked about. So you can kind of turn off which things came in through biocuration, which things came in as direct submissions from scientists, which things are from mobile applications. And we also hold BN trees as well, I should mention here. So BN plants, I should say. So we have a whole set of data that you can pull in and also filter when you're doing your searches. Irene's going to go into detail and kind of showing you how to search data and move into the analysis component of the framework. So I'm just going to give that view here and just show you a couple examples of how to search information out of CartograPlant. So for example, one of the more common ways somebody may come in to search this information is they are aware of a study and they want to pull up that information. So for example, here you can put in your query, study title, put in that title, pull up that study, and look at kind of the detailed view of that there. When you are looking at that information, you're also going to be able to know, are you working with a plot? Are you working with a site? If there's multiple plants at one location, you're going to be able to see that information at this point. You're also going to be able to expand that out and learn more about where that study came from. You can also be searching this information by using the accession number that is generated through our TIPS workflow. So we call these our TGDR numbers. So for example, TGDR 603, we're able to see information, search that particular study and view that on the map. We can then zoom in. There's kind of a detailed view of the plant behind that. And then if you select that study info button, you're able to actually see, okay, I can download the files. I can see how many markers were used. I can see what type of study was used. And we can see that that information has also been mapped on to ontologies as well in terms of the phenotypes. So here's another view. You can also search by genus. So here we're looking at Zia. So we're able to put that in as our genus. We're able to zoom in on a particular study. And here you can see that you have a design across two different plots. And then you could bring those plants in for analysis. You can find out more about the study and so on. Another view here, you could search by genus. Another genus, here's Acer, so looking for maples. You're able to view that here on the map. You could zoom in and look at different studies. And I should say also that you can build queries right here. You can look across multiple studies. You have Boolean functionality here where you can build based on what you're looking for. You can also be searching by particular traits, by particular marker types. All of these things are an option here on the search. Again, here's a view looking at a particular study accession. Again, looking at a TGDR number. We're working in, so if we were looking kind of within our Acer or Maple studies, then looking within a specific study, and because this one actually came from Tree Genes, you have the ability to go back out to the record, either in CartograPlant and sort of the study view that you saw previously, or view it actually within our database here, which then links back out to CartograPlant as well. And here's the other side that I was just saying. So you can look at it on that side if it's a tree, or you can look at it like you can look at all plants within CartograPlant with the view that's available here and access all of those files as well. And so the next thing I wanted to just mention is the whole point of the ability to query studies, group studies, select the plants that you're interested in is the ability to actually perform meta-analysis. So we're taking all of this metadata in so that we know which genome was used, so that we know, was this an assay? Was this a GPS type study? Can we, align all the markers to the same reference before we actually perform our analysis? The analytical workflows that you're going to hear about today will kind of let you know what's possible based on the data that you brought in and the metadata as well. So it will say you've got overlapping phenotypes or you have genotypes that are shared across these studies. That meta-analysis is going to be possible or not possible. And so the engine that runs that actual analysis is the Galaxy framework that enables that to happen. And so this is sort of the whole ecosystem, starting with all that searching, going into the analysis frame and customizing what type of analysis and filtering you want to do on that data and having that all run on the Galaxy backend. And so you'll be hearing a little bit more about that from Irene as well. So in the end, our goal is to integrate relevant environmental layers from curated public sources, provide a mechanism for data to be imported in a fair manner, have HPC, so high performance computing supported workflows that are user accessible and reproducible. That's where the Galaxy component comes in. And then in the future, we're still very interested in bringing in more regional and higher resolution layers. So reach out to us if there's things that you're interested in seeing that are not there. We're adding more functionality in terms of and analytics. So actually providing population structure estimates in real time, things like this on the map. We have that partially implemented, but we've got more work to do there. And then we also generally want to respond to community needs. So if there's things, if there's analytics, if there's data sets particularly that you want to see that are not there, there's always more data to be added. We'd love to hear from you on how to do that. And with that, I'm going to turn things over to Emily to talk to you more about the data submission side.
Okay. So I am going to be walking you through tips. Jill's already given a brief introduction. There we go. But just to go over a little bit again, TIPS is the triple plant PubGen submit. That's the name of our module, TIPS, because that's quite a mouthful. And I'm going to be walking a little bit through background and sort of why we have this submission pipeline, and then walk through the steps that you go through when you make a TIP submission, and show you sort of what some of the different options are, because our studies have a lot of diversity in terms of what they contain: genotype, phenotype, environmental layers, environmental data or layers, and combinations in between. So Jill already showed you this, but TIPS is designed to both take in data files as well as the relevant metadata. And hopefully from the overview of CartograPlant that Jill has just given, you can sort of see why this metadata is so important, right? Data sets that do not have their metadata available, it is very difficult to connect across the data sets, especially when they come from disparate sources. And the whole idea of CartograPlant is to bring data from disparate sources in and make them connectable and usable in these workflows. I'm going to be talking to you specifically about the TIPS submission pipeline for submitting directly to tree genes and CartograPlant. But as Jill mentioned, we have data coming in from TreeSnap, and Meg's going to talk about environmental layers. So these are all, like, the individual studies are from disparate sources. They're from different labs, but also we're bringing in all of these completely different data sets and integrating them all in CartograPlant. So why do we need a submission pipeline? Well, mostly because the data is coming from different sources. So here's just an example of some of the various formats that the data that we deal with can come in. And in addition, so if someone just sends us these files, it is a lot of work to get it all formatted to talk to the other data sets. And in addition, if we just get the data files, we do not have the metadata that allows us to really connect these data points. And then just a little bit on the numbers. So the numbers I'm showing you are a combination of the direct submissions that users have come in and used TIPS to submit to us, and the curation team. So our amazing curation team, they also use a modified version of our TIPS submission pipeline. We make a few modifications because, as Jill mentioned, if you're not the original creator of the data files, it can be quite difficult to come by some of the metadata that we're asking for, which is why we really encourage people to submit to us. But the undergrad team has done an amazing job curating and submitting these studies. So hundreds of studies and over 350 species are represented in these TIPS submissions, which is an incredible amount of variation, right? There's 300,000 plants, and they represent over 1.5 million phenotypes and over 1 billion genotypes. And I started, I don't know, maybe 10 years ago, Jill. And the amount of data, that has come in since then, and the work we've been able to do on CartograPlant to connect this all is just really incredible. So now we're going to do a TIP submission and all of the steps that you need to take from start to finish, basically. The first thing that you need is a user account from TreeGenes. It's super easy and simple. If you request a user account, basically you just need your name and your email. We approve you, and then you can log in. And when you log in, you can go to data submission. And we actually, in addition to the tips pipeline, we also take in genetic maps. So first you select what kind of submission you would like to do. And because this is all connected to your user account, if you start a tip submission and you don't finish it, you can always come back and finish it. Your user account, when you're logged in, you can select from the menu and say, I want to finish this submission, or you can start a new one. And as Jill has mentioned, we have done a lot of work both to collect all of the metadata that we need to make these data sets useful and fair and reusable in CartograPlant, but also to make this as easy as possible for users. So we're going to walk through a few of the different options. I'm going to use one example study, but we're going to break away and look at, a couple different options, depending on what data types you have and your study design. So I picked this study because they had really great, well-formatted genotype and environmental data with great descriptions of it. So it was super easy. I basically just downloaded, they had it on Figshare. I downloaded it and was able to submit it, which is awesome. So you start your submission, and the very first page, you're gonna do your basic information about your study, like your title, the authors, etc., the species that you're working with. From there, the next step is to fill in information about your study, and depending on what kind of data you have, then we sort of branch out and ask you only about the questions that are relevant to your particular data set. And then you upload your files, review everything, and submit. So this is the bottom of that first page where it's asking about your species. If your species is not in the database, you just enter it. It is automatically created in the database for you, as long as it's a plant. There's no work you need to do on that end. So now we're going to talk about the different study design options. So there are drop-down menus for both what data you're working with and your study type. So here's the menu for data type, and you can see it covers basically any combination, genotype, phenotype, and environment. And then the study type also has a wide variety, and really depending on what you select, we're going to ask different questions. So this next screen is going to be the questions that you see if you have a common garden experiment. Because it's common garden, people often do a lot of controls in different locations. So we ask you, what your irrigation regime is, what the biotic environment is like, all of that kind of stuff. But for example, if you have a natural population study, that's not really relevant to you, right? You're not irrigating. So if you select natural population, we're not going to ask you those questions. Okay, so step is to submit your if I say tree accidentally, just apologies, because that's what we started out with, but your plant accessions. So this is the file that contains your plant IDs and your coordinate information. And as Jill mentioned, it does not have to be GPS coordinates. It can be even county, right? And it does not have to be exact. Store all of that information in. So once you upload your file, this box pops up, and we're basically asking the user to confirm what information each column contains. And once you have done that, you can click on this lovely little button and your plants appear on a map. And you can visually confirm that they are in the right spot and not in a different continent or in the middle of the ocean, any of that. So hopefully, this is a pretty easy to use, but also we're collecting this information and getting human confirmation that the information is correct, right? So we're over actually to a different study with phenotype. So for phenotype information, what we do is consistent with the MIAPI, the minimum information about plant phenotype experiments. So we are pretty carefully collecting information about what you observed, what your structure was that the observation was on, what the units were. See it there, what the description of the phenotype is, how it was created, and all of these get mapped to ontology terms, to standard connectable phenotypes. And we are actually in the next few weeks rolling out something to make entering phenotype information even easier, a drop-down menu to select standard phenotypes. It's not quite live yet, but it's almost there. So with a drop-down, if you have a phenotype that's already been entered into our database, like height, for example, you just select that and you don't select anything else except perhaps units, since that does vary depending on your plant. But we're trying to make this as easy as possible and as standard as possible. So because we assign these to ontology terms, when you get to CartograPlant, everyone who has the diameter at breast height phenotype, that is all going to be connected because it's connected to the same ontology term, versus when people upload their files, they might name this all different things, like DBH, or they might spell out diameter breast height, or DBH, 2020 or whatever. And in that case, it's very difficult to connect these phenotypes across studies, but because we use the ontology terms, they're all connected and available for you. And after you complete entering what your phenotypes were, then you can upload your file. And then I want to say, for if you have environmental data that you have recorded yourself versus environmental data that comes from a layer, the loading of that is similar to phenotypes as well, similar to this step. So after we've done our phenotypes, we do genotypes. And we do ask quite a few questions about how these markers were derived. And again, that is to ensure that we have all the metadata that we need to make this useful, particularly in CartograPlant, so that we can do these super cool analyses that you're going to see after the break. So what kind of marker you have, how it was created, a reference for it. So the reference, we're going to see this drop down here. We have many references already loaded. So if they're already loaded in the database, all you have to do is select that from the dropdown and then move on. We can also take in a URL or a GenBake accession, or someone can upload it themselves. So again, we're trying to make this really as easy as possible for users, while also still making sure we have all the information we need to make it useful. You can submit an Excel file or a BCF, depending on what you have for BCF files, because they're often quite large. It does take a little time, but it will go through, hopefully. And we have a little button down there at the bottom to pre-validate it, to sort of make sure that everything is good before the submission goes all the way through. And there's also an option, it's not required, but an option to upload additional files, like a SNP association file. And you can see that we collect information, if you have that, about your SNP association, including what analytic tool you used. And then down below, you can see a few more options for files that we would love to have if you have them to share with us. But again, they're not required. And finally, the environmental layers. So Jill talked about the hundreds and hundreds of environmental layers we have loaded. And if you have used environmental data coming from one of the layers that we already have loaded, instead of having to define and load a file for that, and simply select which data, which part of the environmental layer data set you used, and that information is automatically associated with your plants. So that's the whole submission. You click submit, you get this page that shows you a summary. It goes to us, we approve it, and it gets loaded. And this is the user account view. You can see your submissions. On top are the submissions in progress. Down at the bottom, that's your completed submissions. And then over on the far right, you can see that the status for this submission has been approved. So it's now public. We're going to click on that, and it's going to bring us to the tips view. So this is the view that every single tips submission gets. DOI, you get a DOI, a permanent identifier for your study. You can use this in your data availability statement. These files will always be here and live on as long as TreeGenes does, forever. The tips detail study, tips detail view of this, it has a little map view, and also you can see down below tabs to view information about the study and what kind of data it contains. So we're going to click on over to the genotypes tab. It's a preview of the genotypes in the study. And you can literally go through all of them, but really it's mostly for a preview. We're going to click over to study details. And this gives you the title and the authors, what data type it's got, and also direct links to download the files that were submitted to us. So anyone can come and download it, just like Dryad or Figshare or whatever. But again, the power really in this is to have your plants appear in CartograPlant. So there's a link right up there. And when you click on it, it opens CartograPlant for you, and the study is already pre-selected. And as Jill said, you can add additional parameters. So you can then say, I want to see all of the trees you have of the same species, or I want to see this other study as well. And those will appear in the map. But from there, I'm going to let other people tell you about CartograPlant. So thank you so much to Jill and our tips team. And of course, our amazing curation team for bringing in so many of these studies. Oh, and I want to say, if anyone has any suggestions for how to make it easier for users, or things that you think might be appropriate to submit, but doesn't look like we take them in right now, please contact us, because we would love to hear it.
Thanks, Emily. I'm going to transition over to talking a little bit about TreeSnap, which is a mobile application. I'm Meg Staten. I'm from the University of Tennessee, so moving locations as well. I guess we're all on Zoom, but physically located in Tennessee. And let me share my slides. Okay. Okay. Yep, so today I'm going to introduce TreeSnap. This is a citizen science tool, or it started out as a citizen science tool, and I'm going to tell you how it's morphed into like an actual research, like a research lab tool. New technology has really played a huge role in citizen science, and just thinking about having a device in your pocket that is a computer with a camera and a GPS locator. Anyone can do science with that and can really help researchers. And a lot of us really care about plants and trees that live on the landscape, and they're out there in the wild, and I can't hire enough graduate students to go explore every acre in the whole world. But there are human beings everywhere, and a lot of them really care about science. And so many of you might be familiar with iNaturalist. This is a really great citizen science platform. It's a free app on your phone. And if you go there and you type in your species of interest, there are probably lots and lots of tags and people who have found that somewhere in the landscape. But we have found this to be a little bit weak on the science side. Is the data being collected informative and rigorous? This can be a bit of a problem. I'm super supportive of like K-12 students going out and like tagging things, but it's not always very accurate. So you have to do a lot of curation. How much of that is really being used scientifically? Is it making it into scientists' hands? And so the question we were really asking is, how can we improve on this process? So we found a TreeSnap, which is really a much more targeted attempt to help scientists get information about trees that they need and want. And as Jill and Emily both mentioned, we're definitely thinking about expanding to other plants if there's interest. So if any of you see this presentation and are like, wow, I really need this for my plant, please let me know. And so we're really looking for tree-related research programs to start, just because that's kind of what my lab does, to think about how to collect really rigorous data. So I want to go ahead and do my acknowledgements up front, because this is definitely not a small effort. On the extension and outreach side, we had Ellen Crocker, Bert Abbott, and Dana Nelson. Ellen was really the superstar here. She works at the University of Kentucky and did just tons of outreach to get the platform off the ground and figure out what do we actually need to collect and how are we going to get people engaged. Also on the UT side, Bradford Condon and Abdullah Al-Saeed were our original developers, and now we have Noah Caldwell, who has taken over and is doing a fantastic job. Our original funding was through the NSF, and our current funding is through the Nature Conservancy. So thank you to our funding sources as well. So our first partners were really restoration tree breeding programs. So if you think about going out in the forest health is a real problem. Lots of trees are dying, and the Forest Service in particular and some private foundations are working on breeding trees that are going to be able to live with invasive pests and pathogens. Examples might include the American chestnut, very much a story lots of people are familiar with. Right now, hemlock woolly adelgid, looking at ash trees and the emerald ash borer, which is something my program cares about. All of these have long-term, short-term, or just coming online tree breeding programs. And to start those programs, you need to find the rare individual out in the wild that is resistant to these insects or to these diseases. And this is how the American Chestnut Foundation used to do this. They had a paper form that you had to print out from their website, and then, you know, maybe they got latitude and longitude. Sometimes they just get an address, and people filled this out. And one of the big problems is people don't remember to print this out and take it with them to the field. So you're going on a great hike in the Smoky Mountains or somewhere else. Like, did you remember to print this out and take it with you? Probably not. So we felt like this could be improved on by having an app on your phone that would allow you just on the fly to be like, hey, look, I found an American chestnut. I really want to tell someone about it. So we built the app and we launched it in 2017. It has been up and running for 6 years. So we're very proud of the work we've done, and it's quite stable at this point. It's been through enough enough hands and enough phones that we feel like it's really very stable and mostly bug free. It is available for free on Android and iPhone, so you can go download it and register an account right now. And so while we started with restoration tree breeding programs, we actually have a lot of different types of partners right now. And so when you open the app, you'll see a bunch of different species relevant to your location, and you can read more about like what the use case is. White oak, for example, with the University of Kentucky, they're setting up a new breeding program, and they wanted oaks to be submitted for that. So all different types of purposes. So just to mention all of our partners: the Forest Service, the American Chestnut Foundation, Hemlock Restoration Alliance, Forest Restoration Alliance on the hemlock side, and just so many different people, the Forest Health Group at Kentucky. All of those have been great partners. Key features that will make this app really like better than some of the other things out there: you do not need to have cellular coverage. And I have a picture from the Great Smoky Mountains where there is no cellular coverage in the entire ginormous park. And we can very accurately tag trees, and you can see that the GPS works really well as we're hiking down a trail, collecting trees as we go. Those GPS coordinates are really good. So your cell phone has a GPS chip in it that will talk to the satellites, even if you don't have Verizon or T-Mobile or AT&T. So that's really good. And basically it stores the information locally on your phone. until you come back to the office or go back home, and then you can click the upload button and upload everything at once you do have cellular coverage. Well, I'll say this works internationally. We've had people very successfully use it in South America, in Europe, and Africa. So that's been really helpful for some of our international partners. And we do enforce a high level of privacy. Some of these trees, especially if they're highly endangered, are very valuable. So someone who comes to the website, even if they've registered an account, what they're going to see is our very scientific term, fuzzified coordinates, where we've randomly moved those coordinates within like a 1 to 5 mile radius. And then as a scientist, if you come to me and you say, I'm a scientist, I'm going to use this data for science, we will give you the accurate coordinates. And so when you log into TreeSnap, you'll see the accurate coordinates. And so at the bottom here, this is just an example of what the general public would see. And then if you actually logged in as a scientist and had that kind of account permission, you would be able to see that these samples are actually quite clustered. But the fuzzification process had made that original location wasn't obvious to the public. We also don't share user email addresses or advertise to you or do anything like that. You will have to share your location data with TreeSnap, but we only collect it when you're snapping a tree. We never sell your information to advertisers, and we never use it for any other purpose. We have 2,253 active users, which are people who have actually tagged at least one tree. I think we actually have more like 16 or 17,000 people registered. A lot of people register and never tag. So we're kind of counting the people who've actually gone out there and actually tagged something. And they have tagged 16,636 trees. So we're really excited about that. Ash and chestnut are probably our 2 biggest species. And then a lot of people just really enjoy tagging other things. So I am going to give you a live demo, but as I go through, I'm going to tell you about the 2 projects I kind of want to highlight for this group, which is more of a group that's probably collecting directly for science, maybe less with citizen science. Our ash DIOEC project, we really wanted to go out and find ash trees that were and kind of phenotype them as male or female, and bring those back in and map the sex locus, which is really helpful for the breeding program for ash. And then we also have a genetic diversity project in the Great Smoky Mountains, where I was working with a team of undergraduates. We had them, when they were collecting, include pictures of the stems and of the collecting envelopes. And I'll show you what that looks like to kind of make their collections more robust for research later on. And for this, that was this summer, and we were getting accurate GPS coordinates down to about 4 to 5 meters at most sites. So we felt really good about the GPS accuracy, even in like these really remote locations. Okay, so now we're going to do something really fun where I attempt to show you my phone screen. It'll probably take a few seconds to work, but bear with me. It's thinking. If anyone has Apple products, there's this great screen mirroring that you can do, but it does take a second. Can everyone see my phone screen?
Yes.
Looks good.
Okay, good. Because it definitely takes over my entire computer and I can't see Zoom either. So let's see if we can get to my actual phone. Okay, here we go. So up here at the top left is the TreeSnap app. It's got a little tree on it, so it's pretty recognizable. When you log in, it's going to show you a list of trees. These are going to be trees of interest in your area. So if I flew over to, or it's going to prioritize by trees of interest in your area. Obviously, we're not growing cacao in Tennessee. But at the top are a lot of species we would have in our forests. If I flew to Oregon or Washington State, I might see more like Pacific madrone, Oregon ash. So these should adjust to your location so you kind of know what scientists are interested in. I'm going to select an ash tree and just kind of show you, when I was talking about making this a little bit more robust than iNaturalist, this is what I mean. We're going to ask you for a lot of information about the tree that you're looking at. On the right, I do want to point out there's an information tab at the very top right. If you click on that, you can learn a lot more about how to identify ash, where it's going to be found naturally, who wants this information, so all sorts of good stuff. But going back to actually tagging an ash tree, the first thing it's going to ask you to do is to take some photos. The next thing you can do is select the species. We have lots of ash species around here. You can always click uncertain. We ask you to tell us about the habitat that you're in. Some of our research programs are really only interested in native trees, not trees that have been planted, seeds, flowers, and then very specifically to ash, do you have emerald ash borer? We want to track where it is. We want to track if your trees are starting to go through the process of dying from emerald ash borer. And for this, we do have a show examples link. So if you click on that, you can see the very extensive galleries, what the insect looks like, what the D-shaped exit holes look like. So all of that good stuff. So it's trying to guide you through answering these questions, which is with as much rigor as possible. You just continue to work your way down through this form. As you're working your way down, you'll see at the very bottom, it's refining your GPS accuracy. Mine is at 21 meters. This is not great. This is because I am inside a building. So we have found that when you're in the buildings, it really doesn't work that well. But as soon as I step outside, I'm getting more like 5 meters of accuracy. So that's really great. Another thing that I wanted to show you that really makes this pretty useful for science is this tree identifier option. So I'm going to click on that and it says enter custom ID. So sometimes we have, if we're collecting physical samples, maybe it's called ABC 123 and I'll type that in. But also I can pre-label tubes and envelopes and go out in the field and scan the barcodes. So I just have an example of a little Sharpie package on my desk and I'm going to scan this barcode and it says barcode found. Do you want to accept this barcode? And I can say yes, and then that will be part of my record forever. And I think scanning the barcode actually caused the screen mirroring to fail. So let me try that again. The barcode scanning works really well out in the field. You can create QR codes. You can create like almost any type of barcode you can imagine. It works really well with this. And let me get the mirroring going again. Okay. Okay, so back at the barcodes. And then advanced options, you can mark your comments as private or your observations as private. So if you do not want to be sharing your information, you do not have to. We encourage people who are doing this as citizen scientists to share observations, but it's absolutely optional and totally up to you. So when you're done with all of that, you can click save, save all your entries, and it will give you the option. If you have cell coverage, it will say, do you want to upload this information now or not? And if you do, you can. If not, you can wait until later. I'm going to leave this and just going through, you can go back through your observations that you have collected previously. So I was out in the Smokies collecting things pretty recently. And so I can go back and see, this is an example of an almost dead tree I found. It had a stump sprout with a few leaves that we grabbed. And I took pictures of the leaves and the bud scar for species ID. And just gives me a lot of information about everything I've collected. And if I need to go back and edit or add anything, I can do that. It is going to sort, you can kind of sort this by distance to you, by species. I can also go to a map and see all the observations that I have ever made. This is only going to show you observations for the account that you are logged into. It doesn't show you everything from everyone else. And that is most of the important stuff on the app. Okay, let me get out of this. Okay. Next, I just want to show you a little bit on the website side. Here we go. This is the TreeSnap website. We do have some of the American chestnut folks competing for the most observations. They're very prolific. And you can kind of go check out observations. But what most people want to go to is go see the map. And this is where you can actually see all of the observations from everybody that are public. So it takes the map a minute as you zoom in and out. It'll try to aggregate the observations into little points. So you can't, otherwise it turns into so many points you can't see. So give it a minute when you're zooming around. And let's start. We'll go to where I am right now. Yeah, so as I'm zooming, and all of these different colors are different tree species. And We have tagged lots and lots of trees right around my office because we add a new feature and then we walk outside and we go test it. So this is where I am right now. And so I can see all of the trees that I have tagged. Let's find one that we actually tagged for our project. Okay, so here's a great one. We put in our custom identifier. We put in the comments Badash Collection 2021 because that was the name of our collection expedition. And I can see all of the information. And if I go down here, I can visit the observation page. I can see that I was the one who tagged this one, all of the different information I took, and I can go through and look at all of my pictures. And one of the reasons this is really great for research is that I can get a really nice view of the flowers that we collected. And so let's say this pops out my analysis is not fitting the trend of everything else. I can go look, maybe we mis-ID'd it. Maybe we collected, maybe we called it a female and it was actually a male or vice versa. So just really convenient for me to be able to go back in and like actually get back to the sample and see what we collected. Let's see. Let me go back. And then just quickly, I'll also show you some of our Great Smoky Mountain samples and how I can filter into those. The Great Smoky Mountains are very large. It's a very large national park, and we were collecting ash. So the blue is ash and the green is American chestnuts. The American Chestnut Foundation has also paid a graduate student to just hike through the Smokies and find American chestnut, which sounds like a great summer project. I want to go back to grad school and do that. But here are all our samples from this summer. So we were really pleased with, we did some car collecting and you can see the GPS really follows the road very nicely. We did some trail collecting. Again, those GPS coordinates are very accurate. I can click, I can see the full description. Esmeralda is one of our undergrads, so I can see what day and time, and she brought us some pictures so that if we ever need to go back and try to find this tree, we can do it. If we have questions about whether what she collected was the right sample, we can see the actual envelope that she put the sample into. She takes a picture of it. She takes a picture of the leaves. So we just get lots of information for follow-up later on. Again, the bud scar is very useful for species ID. Let's see, go to the admin. This is just kind of an administrative dashboard. If you get registered as a scientist, you will also be able to use this. I don't want to take too much of your time, but if you want to use this platform and you're a scientist, you can add users from your group. You can add citizen scientists, you can add your graduate students and postdocs, you can add anyone you want as a user, and then form a group to track their observations so that you can filter and grab all their observations at once. You can go through and filter for observations and click if they're the right species or the wrong species. So if you need to do some filtering because you do have some citizen scientists or some bad data, you can do that. You can keep collections, you can download everything in Excel spreadsheets. So lots of really nice options on the PI side to manage your data, to get your data. And of course, all of this also automatically gets exported to CartograPlant. So if you're using CartograPlant, you don't actually have to do anything. All of this will magically show up there. And that is super convenient. So that's the recommended way to do it. Let's see, I'll go back to my slides really quick. Yep, we're super excited to partner with anybody. We'd love to expand out into outside of trees. We just had a lot of success with trees. We're working with more groups who are doing lab collections and not citizen science collections. So we've really learned a lot about how to make TreeSnap really useful for that. So contact me if you're interested in that. I don't know if we have questions, but I will, I'll let Jill address that. I will say thank you to everyone who worked on this app and to the CartograPlant team for working with us and importing our data.
Thank you, Meg. Perfect. We had a couple of questions in the chat, so I've addressed a couple of those, but you might want to respond to a couple others as well there. We do have a break now, so we'll actually come back in exactly 10 minutes. And this is where we're going to get a little bit behind the scenes first with Rish on the CartograPlant environmental aspects. And then from there, we're actually going to jump into the analytics of CartograPlant for GWAS. So we're looking at association mapping, landscape, genomics, looking at population structure, filtering data. How do we actually take everything that you're searching out of CartograPlant and run it into an analysis at that point. So if you have questions, go ahead and put them in the chat during the break, and we'll try to help you out with all of those. We'll also have time for official Q&A at the end, but keep them coming in the chat as well, and we'll address all of those as they come through. And we'll see you in now 9 minutes from now. So see everyone shortly. Okay, I'm going to go ahead and stop my share now. I'm going to turn the floor over to Rich, who's going to tell us a little bit more about CartograPlant.
Awesome. Thanks so much, Jill. All right. So hi, everyone. Just give me 1 second. Let me just hopefully get this. There's so much windows here. Okay. Hi, everyone. Thank you for the opportunity to present to you behind the scenes of CartograPlant. My name is Richard, but most people call me Rish, and I'm the lead developer at TreeGenesDB and thus CartograPlant as well. I'm super excited to share with you some of the technical aspects of CartograPlant, in particular, the world of map layers and the technology that creates the CartograPlant experience. So just before I begin, I want to thank the entire TreeGenesDB CartograPlant team, since this would not be possible without them, and of course, Dr. Jill Wegazin for her leadership and direction on this exciting project. So let's get into it. Today, we'll be touching swiftly on the following sections. What are layers, very briefly, the types of layers that I've come across working on CartograPlant, how we work with contributed layers and data, how we get about serving those layers to the user. And at the end, if you have any questions, I'll try my best to, of course, answer them as well. Just before we get into the technical aspects of layers, we've compiled over 900 layers into CartograPlant and counting as well. We've tried to organize them into groupings to make it easy for you to find a layer that you might be interested in for your research. From forest fragmentation to species range maps, climatic variables to density populations. We have some basic statistics as well of what gets accessed for the year on our CartograPlant website. And on the graph on the right, you will see that the mean annual temperature, forest fragmentation risk, tree cover, major soils groups, and mean annual precipitation seem to be the most popular layers for this year. It might actually been a little bit late last year. And of course, today, we're talking about the layers and data and how it works in CartograPlant. So let's get started with what is a layer. I'm pretty sure most of you know that, well, what a layer is on a map. You might have used like apps like Waze or Google Maps and many other services that use GIS layers. So you've probably seen or used one of them, hopefully. But if not, skip all of that and just go to CartograPlant. We think about these layers just as it is. It's an image placed over a map. And when you think about it, a map is also an image as well. If we were in Photoshop doing some photo editing, that would be accurate. However, a GIS layer goes a little bit further, if not a lot further. For example, a species range map, such as the one in green, this layer represents a geographic region. In this case, a large subsection of Canada, as you can see there, hopefully, and is therefore based on boundaries, while the purple-pink layer represents individual trees on our CartograPlant map, which of course are based on minute points on the map. Both examples that I just presented to you are geo-referenced data. It's an example of geo-referenced data. It's just a big term to refer to a geo-coordinate referencing system, which also sounds big, but basically it's pointing to latitude and longitude. I think it's obvious that one is showing a range, the other one is showing individual points. So the range uses polygon shapes in the background, which is a little more complex to define, while the points on the map are single coordinates and are fairly easy to define, right? Because you could define those as just latitude and longitude points. In the previous example, both layer examples were vector formatted layers. So vector data is usually made up of vertices and paths. They are calculated mathematically, or they have a mathematical representation. So what we tend to see in vector layers are polygon areas, example, the range map that we saw, leftmost map on the left, and also the points, which is like the middle map that I screenshotted there for you guys. On the other hand, some of our environmental layers, such as the world climate layers, are what you call rasterized layers. They are made up of a grid of pixels, just like normal non-georeferenced images, such as photos from a camera. However, rastered layers usually have georeferenced data, which are used to position them accurately on the map as well. So you think about a map, a rastered layer like a photo, but that photo has some idea of where to be placed on top of the current map, the world map that we see. So why two different types of layers? This slide summarizes the advantages and the disadvantages, which are basically inversely proportional or inverse points, so to speak. Vector layers, being mathematical in nature, they tend to be smaller in size compared to rastered layers. The mathematics generates them on the fly. So when you're loading the actual map and you're zooming in, zooming out, your browser is actually doing that. While the rastered layers are not very mathematical in nature. Consider them to be pre-rendered, and thus they take up much more data. So a vector layer, for example, could be 500 kilobytes in size. You know, a long time we could probably compare that to a floppy disk. It's about half the size of a floppy disk. While it's not unusual for the rasterized layers to be 10 gigabytes, which is like tens of thousands of times the size of a floppy disk. And this leads to natural opposites in how they are processed by the user interface. The user interface that we generate, which is the map, we actually use a library called Mapbox to do that. And vectors usually need to be rendered on the fly, and this can be more CPU intensive, usually because the user's computer will have to render that part of the map, while the raster layers are mostly pre-rendered, and so they can be quickly accessible, assuming that you have the bandwidth speed to be able to retrieve those images fast. A perfect example of this can be seen in CartograPlant, where we use vector type data to display tree snap trees, the purple tree points that you see in the screen on the slide here today. And we have about 16,000 trees, so the loading and rendering time isn't terrible. It's about 2 to 3 seconds. But if TreeSnap were to go, if it were to expand to maybe 100,000 trees, we'd have to think about converting that layer to something more speed efficient. When we had originally tried the vector method for importing 1 million trees from the BN dataset on your right, the load time was more than 30 seconds to a minute, and nobody wants to wait like an entire minute for a map to load. And this is the whole reason why I'm kind of explaining the difference between the vector and the rasterized. So what we ended up having to do was we ended up having to rasterize that data into something called an MB tile format to serve those 8 million trees instead of 1 million trees. So I think we broke the barrier there to some degree. We use Mapbox.js, which is an open source library to render these layers into the browser. In case you ever want to contribute, of course, to CartograPlant or come across these formats in useful papers, please let us know as well, and we'd love to add them in. The formats with the asterisks at the end are preferred since these work well with GeoServer, which is what we use as our tile server for CartograPlant. So from CartograPlant's perspective, Map.js, the open source library, renders the map to the user, but the map has to come from somewhere. It has to have its own source. It's not just a file, even though the layer file is the most important part of it that has the actual data. The file has to actually be served by a tile server. So GeoServer is an amazing open source tile server that we use on the back end to serve our layers to the user, the UI, which is the map. A future slide will show the workflow in a little more detail when we summarize to the end. There are many other georeferenced formats out there. And so what we end up doing is that because there are other formats, if there are other formats, we usually have to convert them to these easier formats, which are compatible with the GeoServer, our tile server. But from this list, the original 1 million trees from BN were initially formatted into GeoJSON vector format and then converted into MB tiles, a rasterized layer, using something called Typicono. It's an app called Typicono, using the Mapbox recommended guidelines. So the Mapbox recommended guidelines, they explained how to go about doing that conversion for us from vector to rasterized. And this made the loading of the 1 million trees bearable and efficient, since it compiled an MB tile layer, which could then be used by the tile server, which is our GeoServer. In other words, it's not absolutely a simple process. Sometimes the format has to be right for it to work properly. A lot of the time was spent manually converting these public layers, which were in different formats, into compatible formats that we could use via GeoServer. So as you can see as well, CartograPlant leverages a lot of open source technologies. Plus it uses our custom open source code as well to glue things all together. So how do we get layers into correct types and formats? How we work with these layers? You surely need powerful software to do this, since since visualization is an important part of the layer sections. Discovering QGIS and learning that it was free was a lifesaver, and a very good example of how an open source community can create powerful software. With QGIS, you simply right-click on a layer you've opened, and then you click export, and then you can select a new coordinate system to convert and the type of format you want to convert, and it does all the rest for you. And this is what we use. So we use this app this software to convert from one format to the other. The great thing about QGIS as well is that it supports the vector, the raster, and even other types of hybrid layers as well. And it even tries to pull layers from online servers as well too. Under the vector layer, as you can see, it's a long list, and I am pretty sure that there are others supported in QGIS that I didn't even touch that was on this list here today. The same for the rastered layer file formats as well, as you can see from the screenshot. So many different layers, many different support, and it's free. I know I mentioned that GeoServer is a tile server. I just want to explain that very quickly because it's actually quite amazing what is happening in the background. So serving a single file would take more than a few seconds to download, which is bad for the user experience. The tile server is able to update the map, for example, when it's zooming in or out, using what looks like grid squares. Because the internet is, so much faster now, you may not notice that as much, but that certain parts of the map are being updated instead of the entire map. A GeoServer is responsible for this calculation and thus efficiently doing that in the background. So multiple tiles are retrieved from GeoServer in order to display a map at any point in a zoom, in wherever you zoom, if you zoom in or zoom out. And this essentially means that the map loads faster on updates because less square tiles need to be downloaded in most cases. And this is another reason why we use GeoServer as a middleware piece of software to do that. Our code handles whether the layers are vector or rasterized, and then Mapbox renders it to the user. I want to show you what the data looks like in the background of all these beautiful maps and points. It's a little bit intimidating at first, but it's actually simple. The vector format, GeoJSON, is a good example of how data can be stored in vector format. This example shows a single point referred to as a feature, which contains multiple properties. If you look very carefully, you might be able to find ID and icon type. I highlighted it, so hopefully you can see that. So it's very easy to add additional properties. It could be species, it could be genus, it could be whatever you want, etc., into this string that you see here, which looks a little bit complicated. We removed additional properties to speed up the load time on our initial load on CartograPlant. We use the ID to cross-reference the database instead, or the database tables that we have instead, to retrieve additional information, such as the species, the genus, all these other information. We have a lot of information out there, so I can't list it out. While this is the easier format, as soon as the data crosses about tens of thousands, you can see performance degradation. In our case, when that happens, we would probably likely switch over, not probably, we would switch over to rasterized layers for that, for those, if it's too large. So we developed this API, application programming interface, that converts the table into GeoJSON format for the initial 3 points. And you see this when the map initially loads. So the data actually comes from the database. It then gets converted into the GeoJSON format that I just showed you. So the data might come from, for example, the TPPS study. In most cases, it comes from the TPPS study that has been submitted through TPPS. It gets imported into a table like the one that you see above, with additional information like the species, subkingdom, and so on. The API filters these rows for only the essential needed data for a GeoJSON object, like the coordinates and the ID of the plant, and then it sends this to Mapbox rendering engine to display to the user as the map and the points. Our database administrator, Emily Groh, had to create special views so we can retrieve this data as fast as possible. And as you can see, the GeoJSON points back to the ID of the data in the table. So the additional information can be retrieved and displayed on the map. For example, when a user clicks on a tree, then it does the actual querying into the table and then brings that additional information back to the user. Now, what we touched was the vector layers. For the rasterized layers, the data properties are stored within the layer files themselves and not usually in plain text. You can't just read it just like that. GeoServer has its own pre-coded API functions that makes this easy for us to extract data and receive GeoJSON as well. So it converts the rasterized data into GeoJSON if we require it. Our code doesn't have to change too much, therefore, to handle data, even though, in essence, an entirely different engine is being used to do that. That's the GeoServer middleware. What you see is a screenshot of the mean annual temperature layer enabled. I clicked on a point on the map, and that's showing hopefully that pop-up there that you can see. This actually queries GeoServer to retrieve the mean annual temperature for that point with latitude 20.828 and longitude minus 100.106. The result is we get a mean annual temperature value of 21.16 degrees Celsius. So all that is happening on the fly in the background. A little bit of an offside, but I think it would be informative to mention, most of the technologies that we use are open source, such as Triple, which was built on PHP, and thus the CartograPlant module as well, which is built on top of Triple, also, and which has the admin interface as well, is also built with PHP as well. Second to this is the background API engine, which uses JavaScript, and also the foreground, the map itself, which also uses JavaScript as well. So it's also built on JavaScript, and meaning that for the most part, you only need to be good at these languages, hopefully PHP and just JavaScript, in order to contribute if you ever feel that you want to contribute. I'm beginning to close up here, but let me just touch on the components again, because CartograPlant looks like a map, but it's more complicated than that. As you can see, it actually takes 4 components for CartograPlant to do all that it does. The CP UI, that's the map the users see, the CP admin, the background control that happens. There's the CP API, which the map pulls metadata from, and also the map API, where layer data is outputted to the map UI. All of these components work with the layers in different ways. The admin organizes the layers on the user interface. The user interface displays the users, the layers to the users. The API pulls embedded data from the layers, and then GeoServer API outputs the layers to the map and also answers embedded data queries as well. We spoke about the map UI and GeoServer a bit. There's 2 components. I just wanted to show you that there's also an admin UI that controls some critical aspects of the map user interface, and we normally use this to manage some of the organization in CartograPlant. I hope that you can see from the screenshot that the admin UI stores the GIS server configuration, the API server configurations, and it is most primarily used to configure the layers available in GeoServer. Firstly, you can group layers here, which would then show up as grouped on the map user interface as well. So for example, the environmental layers, when you have to select those, they are grouped in a certain way. We use the admin interface to do that. In addition, I don't know why this one is not displaying. Right. In addition, you can pre-configure each layer with details and options of which will affect what options are available to the user. So you can configure that from the UI, the admin UI. On the map user interface, for the user to adjust. For example, we add the layer information details in here, and we can even show or hide specific fields from the layers embedded within GeoServer as well. So it makes the map easier. Sometimes when we import data from when we import layers into GeoServer, there's information that we don't necessarily want the user to see because it just clutters the user interface, and it's not really applicable to research. So this allows us to hide these things or show these things as necessary. I left out the CP API because this is a command line tool we use with additional APIs. For analysis and Galaxy, which is a whole other topic probably, we use the CP API very, very closely. But I have to mention that the API is used to run jobs in the background and do some of the automation. All in all, this hierarchy is hopefully easier to understand. At the bottom, we have the raw data or processed data, usually stored as files or in some cases within database tables. GeoServer reads this data and is able to output this data as a layer, which can then be seen on top of the map itself. In between GeoServer and the map, the CP admin controls the categories that are seen on the map, for example, the environmental layers, as I mentioned. Hopefully, this makes even more sense. The CTUI, the map itself, is where the user gets a list of the environmental layers from the admin. When the layer is clicked, it calls GeoServer. GeoServer looks up that file, generates the tiles, then brings that back to the map interface. When a user clicks on a point on the map, it then queries GeoServer again, or sometimes it also queries the database as well, and returns that as a map pop-up on the actual map as well. And there you have it. There was a quick behind the scenes look at environmental layers and data and how CartograPlant components work. Thank you so much for being an awesome audience. And if you have any questions, maybe we could try to answer them. Due to time, maybe during the chat, I could try to answer as well. And back to Jill.
Thank you, Rich. I think we'll move on now to Gabe, who's going to give us a background on some of the analyses that can be performed, so just general background on what those are. And then Irene's going to follow up as last but not least to tell us how to do it in CartograPlant.
Okay, thank you, Jill. So a little bit of background into the data analysis with CartograPlant. But first off, yeah, my name is Gabriel Barrett. I am the analytics workflow developer for CartograPlant. And let's get to it. So in a nutshell, CartograPlant intends on providing users with the ability to perform data quality filtering, genotype imputation, genome-wide association, and population structure analysis. And you will see these specific topics frequently appear in population genetics in general. Well, before we actually dive into the background, I want to share with all of you that CartograPlant performs all of this magic through the use of Galaxy, which is an open source community maintained software. And its focus is to make data analysis more user-friendly by eliminating the need to code via interaction with the GUI. And it hosts a wide variety of bioinformatic tools that are ready to go. So let's get to it. First, GWAS stands for Genome-Wide Association Study, which is an approach used in genetic research to associate genetic variation, usually single nucleotide polymorphisms with the trait of interest. Now, a single nucleotide polymorphism is a substitution of a single nucleotide at a specific position in the genome, and that should be present in a relatively large fraction of the population for analysis. In the bottom left diagram, I'm showing you genotypes for 6 individuals. For individual 1, I have highlighted its genotypes in green and its corresponding phenotypic value in yellow. I want to draw your attention more specifically to the SNPs highlighted in red for all individuals, because this polymorphism has a strong correlation with the individual phenotypes and thus must likely be causing the resulting phenotype, which in this case is a response variable, and the SNPs is the predictor variable. Now, for landscape genomics, these variables get switched, where the predictor variable is now the environmental condition in which we find our organism to be contained within, and the response variable is its resultant genotype. In landscape genomics, we are trying to identify relationships between environmental factors and genetic variation that help narrow our focus to specific candidate sites. I have introduced you to 2 slightly different approaches commonly used in population genomic research. One being landscape genomics that resembles a bottom-up approach, meaning we start from the molecular portion of the organism and try to build our way up into identifying potentially adaptive phenotypic traits responsible for higher fitness levels in those environments. The second type of study was genome-wide association, a top-down approach, where we know the phenotypes and want to explore the molecular mechanisms causing these patterns. There are a plethora of other mechanisms, like phenotypic plasticity, that we won't get into today, but surely play a role in local adaptation. And in summary, these approaches want to identify markers under selection within that environment are causing expression of that phenotype. And like with any raw data, sequencing data has its own sets of errors and biases that need to be evaluated prior to conducting analysis. So the first thing I want to get into is genotyping errors. And a genotyping error describes when the observed genotype does not correspond with the true genotype. And these genotyping issues can arise at sample and library preparation and sequencing. The solution to removing genotyping errors is by using filtering thresholds based on quality metrics. There are many different approaches and metrics for determining genotyping errors, but the most common are based on low quality scores, high levels of missing genotypes, and minimum genotype counts or minor allele count. Sometimes these fixed thresholds can be applied at the per site or per individual level. And in the bottom left diagram, I'm demonstrating filtering per site exemplified with the red axes, and this could be due to not meeting any one of the quality metrics described above. In some cases, we may want to remove entire individuals from the study because they just simply did not sequence well. Like for example, in the bottom right, we're removing individual 4 because maybe they had low genotype quality across all of their sites and are exacerbating quality metrics at the per site level. Next on our list is missing SNPs. Now, before I move on, I'd like to add that the level of missing genotypes can largely depend on the type of library preparation conducted for that study. Even after doing our quality assurance, there still might be a small fraction of missing SNPs retained in the data set. For genome-wide association studies, really any correlation-based studies, missing data can drastically reduce the statistical power for detecting significant association. The solution to this is imputation, meaning we use a model to come up with the best possible genotypes at that particular site. So there's many tools currently available, and they either leverage a reference panel or linkage disequilibrium to compute what that most likely genotype will be. And linkage disequilibrium is the non-random association between 2 or more genotypes in that population. A large principle to this is that the closer the 2 markers are, the higher the linkage disequilibrium. And this also serves as a basis to identifying locally adaptive genes in genome-wide association or landscape genomic studies. One tool that is currently adopted by CartograPlant is LinkImpute-R, and it has 2 capabilities. The first being that it can test different combinations of quality filtering thresholds and will output to the user a data frame in which you can decide on the best filtering combination. This way, you can maximize the quantity and quality of the SNPs used in your downstream analyses. And then the second capability is that you can impute and therefore remove any missing data by leveraging linkage disequilibrium. And this does not require a reference panel, which is often lacking in your non-model species. Next on the list is to talk about how population structure can influence the accuracy of genome-wide association and landscape genomics results. Simply put it, the solution is just to account for it as a confounding variable in the models, which a lot of tools that are being created do that now. But a confounding variable, by definition, is an extraneous variable in the model that correlates with both the dependent and independent variables. Now, population structure is interesting in and of itself because it describes the level of connectivity across the landscape. But in GWAS, this population structure can cause spurious associations leading to inflated levels of false positives in analysis. So for example, in the diagram to the right, population A and population C may strongly exhibit genotypes highly correlated with elevation that might actually just be due to lack of gene flow between the populations and be caused by genetic drift as opposed to selection, which is what we are testing for when conducting these landscape genomic analyses. The last thing on our list is multicollinearity. It's a tongue twister. Oh, sorry. So there are so many environmental layers, which is amazing because we can answer many different biological questions. However, not all of them will actually be informative due to this phenomenon called multicollinearity. Which this refers to is the linear relationship between two explanatory variables. To avoid this, we usually implement a method called variable selection that describes the correlation between two variables and allows for decision-making on which variable is relevant and which should be included in the analysis and which one should be thrown out to avoid any redundancy. And CartograPlant provides this capability through the use of Pearson correlation analysis. And the main objectives of CartograPlant is to provide all of you with the workflows capable of performing genome-wide association, landscape genomics, and meta analysis. Meta and mega analysis is a set of methods that allows the quantitative combination of data from multiple studies. These methods also allow quantitative evaluation of consistency or inconsistency of the results across multiple data sets. The main difference between the two is that mega-analysis combines the raw starting data, whereas meta-analysis combines the final results. Combining studies with mega-analysis can increase the power to detect association signals by increasing sample size and by examining more variants throughout the genome than each data set alone, and is coined the gold standard of empirical research. However, mega-analysis is not always feasible due to the raw data being inaccessible. But this is the ultimate objective of CartograPlant. And here is a diagram demonstrating what we expect the capabilities of CartograPlant to look like in the future. And highlighted in red are the tools that are currently available in CartograPlant. So in conclusion, CartograPlant provides flexible analytical workflows for population genomic analysis, native support for mega and meta-analysis, constantly develop features that follow present-day standards, and finally, a centralized platform to integrate, visualize, and analyze high-throughput biological data. And with that, I would like to conclude my talk and hand it over to Irene for a wonderful demo.
Thank you, Gabe. That was fantastic. If anybody has any questions, keep them coming in the chat while Irene goes ahead and shares her slides.
Well, okay, if there are no questions, I'm going to start my presentation. So as Gabe said, I'm going to present a demo about how to perform a data analysis in CartograPlant. The first step to perform data analysis in CartograPlant is obviously to access the interface, the CartograPlant interface. And to do so, you have to go to the main page of CartograPlant, which is cartograplant.org. And if you do that, you will see, you will see this link here that says get started. And by clicking on it, you will access directly the CartograPlant interface. So as you can see, it is divided into two sections. The first one is the so-called left panel, which is the action panel where users can interact with the plant and the map. So they can select the environmental layers of interest or the plants of interest, and they are displayed in the right panel, which is an interactive worldwide map that displays all these plants and environmental layers selected in the left panel. Another interesting feature for data analysis in CartograPlant is the possibility to navigate through the map. This is useful because in order to select the plants for that analysis, you may want to see the plants in context. And there are three main ways to do that. So users can navigate through the map in the left panel by using this map navigation section and typing directly the latitude and the longitude coordinates of interest. Another way to do that is by using the these zoom buttons in the right panel here. And the last way to do that is just directly, since this worldwide map is interactive, just directly clicking on the map and dragging, and you can navigate through the map. Another interesting feature for that analysis is the possibility to display the environmental layers in CartograPlant, as Jill explained briefly before. But I think it is important also for that analysis because this way you can select the environmental data of interest. You can visualize it and see if these environmental layers are interesting for you, and also to see the context of your plants, so where the plants are located. So in order to display the environmental layers, you have to go to this map options section, click at this button here, and you will see automatically displayed all the environmental layers available in CartograPlant. In this case, I'm displaying the forest fragmentation layer. So there are, well, the environmental layers are classified, mostly classified depending on their location. So there are environmental layers that are local from North America mostly, and others are worldwide. In this case, I'm displaying one from North America, the forest fragmentation layer. And again, the worldwide map is interactive, so you can click directly on the map and display information about the environmental layer at this specific point. So this is very convenient as well for that analysis because you can see the conditions at this location, and if the plants surrounding this location are interesting for you for that analysis, depending on your biological questions, of course. Oops, sorry. Okay, and now I'm going to explain how to select plant for that analysis, which is the key point for this data analysis. So there are several ways that users can do that. One way to select plant for that analysis is by drawing a polygon directly on the map using this polygon drawing button here. So all the plants that are surrounded by this polygon are automatically displayed in this map summary section, and they are available for that analysis. Another way to do that is by clicking directly the icons of the plants and trees available in CartograPlant. So there are tree and plant icons. So by clicking on them, this window is displayed about showing information about this specific plant. For instance, the latitude, longitude, and the elevation, the IV for this plant, taxonomical information. There is another button showing the study info, the information about the study to which the plant belongs, which is also useful to decide if this plant is interesting for that analysis or not. So this is the information basically that it is displayed, like the study title, the authors, and also the data available for this study. And finally, by clicking this Add Plant button, users can select the plant for that analysis. So it will be automatically added to the map summary section to the selected plants for that analysis. Another way to select plant for that analysis is by using these plant dataset sources here. So as Jill mentioned, there are several sources for plants in CartograPlant. Imagine that, for instance, we want to select plants coming from the internal submissions, which is the plants that come from the direct submission of our biocuration team. Then you can select these plants by clicking this button here and just off all the rest of buttons. And as you can see, the number of plants are drastically reduced, displayed in the worldwide map. And once you do that, you can click this Select All button here and select all the displayed plants for that analysis. This is another way to select plants for that analysis. And the last way to do that is by using this filter section. So in this case, you can select the plants using different filtering criteria. Some of the filtering criteria are taxonomical, for instance, the family, the genus, or the species. Also the molecular markers available for these plants or the plant structure, the phenotypes, and the study based on the title, the first author, or the accession. I'm going to show you how to select plants based on the study accession number, but you can also use the other criteria depending on the questions you may want to ask. So by clicking this study accession here, you will see all the accession numbers available in CartograPlant, but a very new feature that we have now in CartograPlant, since we have a lot of studies, is that users can type directly the study ID here in order to select the studies of interest. And automatically the number of phenotypes or the phenotypes available for this study are displayed here, which is also very convenient. In case, for instance, you want to perform a meta-analysis and you want to have overlapping phenotypes between your studies, or you are looking for a specific phenotype, it is very convenient to select or to make sure that this study is interesting for you. And you can add several filtering criteria. So by clicking this add rule, you can add other filtering criteria. And in this case, I'm adding another study accession number. Again, the phenotypes available for this study are displayed. And finally, by clicking apply filter, this apply filter button, the plants are again subsetted. And you can click this select all button here to select the displayed plants for future data analysis. And finally, by clicking this little circle here, you can also see the number of species to which this plant belongs. That it is also very useful for analysis, because depending on your questions, you may want to have all the plants belonging to the same species or analyzing several species. And finally, once you have all the plants selected for that analysis, you can click this analyze button here. In order to do so, you have to be authenticated. So you have to register and log in 3 years. And if you navigate authenticated, you can click this analyze button here in order to perform that analysis. Once you click this button, you will see this CartograPlant analysis window here. And as you can see, it is divided into several tabs. So the first one, that is called manage, it provides a unique analysis ID number to your analysis, but you can also provide an analysis name here, and also select the analysis type that you want to perform. In this case, I have selected genotype by phenotype by environmental data, but you can select any combination of genotypic, phenotypic, and environmental data depending on your questions. And also the users are strongly encouraged to create a workspace. And this is important because in this workspace, all the output from your data filtering and analysis will be stored there. Not only for this analysis, but also for future analysis, which is very, very convenient as well. And another important feature is that you can keep track of the data available for analysis in the top part of the window. So as you can see here, you can see the analysis ID and also the number of phenotypes, genotypes, and environmental layers. Now we have 0 because, of course, we have just started. But you will see throughout the proxies how it is automatically updated, which is also very convenient to keep track of the data that you have for your data analysis. So now I'm going to provide an analysis name, also create a workspace in order to store my output files of the analysis and filtering. And finally, I will click next in order to go to, that will direct me to the next tab, which is called filter by trait. So here, we are going to be able to filter the phenotypic traits available for our studies. So in this tab, the first thing you can see is are the studies to which the selected plant belong. We already know them because we have selected the plants by study, but in case you have performed this selection based in other criteria, it is very, very useful to know the number of studies to which your plants belong, and also the phenotypes available for these studies, and the number of phenotypes available for each phenotypic trait. Another important feature that is available in this section is the overlapping stages of your phenotypes, which is also very useful in case you want to perform, for instance, a meta-analysis and you need the phenotypes in common between the studies. You can select the phenotypes based on these overlapping stages. And also another novelty that we have recently added to CartograPlant is the possibility to change the units of your phenotypes. So for instance, in this case, in plant height, we have the option to transform the units into meters or centimeters, depending on your preferences, which is also very, very convenient. So once you have selected the phenotypes of interest, a PCA is automatically created as a means of data exploratory analysis. And also the phenotypes are displayed in histograms in order to show to see the distribution of your phenotypes throughout the individuals. And these histograms are interactive. So by clicking this adjust threshold button, users can subset the phenotypes depending on the distribution. So depending on their biological questions, they may be interested in the center of the distribution or the tails of the distribution. So once the users have selected the range of the distribution of interest, by clicking this save adjustment button here, they can save this data in the workspace for future data analysis. Then the next section is other filter, but in this case by genotypes instead of phenotypes, but it is essentially the same. So in this section, again, we can see the number of studies to which the selected plant belong, and also the overlapping stages of the SNPs, of the markers, of the genotypic markers. In this case, SNPs. So this overlapping stages is displayed in a Venn diagram, which is very visual, and also here is described here. So users can select these SNPs of interest based on their overlapping stages, which is again very convenient for, very useful for meta-analysis because we want overlapping SNPs between the studies. So we are going to select the overlapping SNPs, of course. And as you can see, it is automatically updated here. So now we don't have 0 genotypes anymore. We have about 24,000, and also the number of phenotypes that I forgot to mention it in the previous slide. And well, another thing that you may ask is that, okay, but maybe we don't have overlapping SNPs between studies because they are mapped against, for instance, different versions of the reference genome. So, but they do exist. So we are working in providing this reference genome information. So saying, okay, maybe these SNPs are mapped against the version 2 and these ones against the version 3 of Populus trichocarpa, which is the species to which these plants belong. And also the possibility to remap these SNPs against other versions of the reference genome. So that they are mapped against the same version, and we can find overlapping SNPs between studies, or have them the most updated as possible by mapping them against the most, the most recent version, so that we can have them updated without the need to remap all the SNPs again, which is very, very powerful as well. So once we have the genotype selected for that analysis, we can go to the following section, which is the filtering and imputation step that Gabe explained to you before why it is important to perform that analysis. So here we're going to be able to perform SNP quality filtering and impute the lacking SNPs. And to do so, CartograPlant requires that the SNPs are in VCF format. So this is why, first of all, CartograPlant checks if the SNPs are actually, from both the studies, are actually in VCF format. And here you can see, you can access to all the SNP quality filtering and imputation workflows available so far in CartograPlant. So you can see the link imputer program that Gabe mentioned that allows this decision of the best quality filtering thresholds for your data. And also the imputation of the lacking SNPs. And also users are allowed to perform this data quality filtering manually as well by selecting the thresholds of interest. So I'm going to show you how to perform a one of these steps of quality filtering. So first of all, you can select here the files available in the workspace. In this case, we are selecting a VCF file for this study. And by clicking here, initiate analysis job, you can just directly start the filtering. It's very, very quick. And The output results are available in the workspace. So here and in the Manage tab. So users, by clicking directly on them, they can download the output files in their computer. And also, in order to help users decide these quality filtering thresholds, we are working in providing interactive histograms, very similar to those for the phenotypic traits, in order to help them decide the best quality filtering threshold for their data, which is a tricky aspect of quality filtering. And also, we have available a workflow that is not actually a quality filtering, but it is necessary to perform population structure analysis, that is filtering by linkage disequilibrium, because the population structure programs assume independent SNPs. So we have here available in CartograPlant. It is possible to perform this SNP quality, this linkage disequilibrium filtering. So we are going to perform it as well. And again, the output files are available in the workspace for future analysis. So the next step will allow us to perform this population structure analysis that also Gabe explained to you why it is important in the association analysis in order to avoid these spurious associations. It is also interesting by itself for population genomic studies. So currently we have available only one approach to do that, which is fast structured program, which is a Bayesian approach. And to do that, users can select the number of populations they want to test, and also the VCF file available in the workspace. So we will select the linkage disequilibrium SNPs, filter SNPs. And by clicking generate fast structure, users can perform this fast structure analysis, this population structure analysis. And once the analysis is completed, all the output files are available in the workspace for future data analysis. We are also working in providing other approaches to perform this population structure analysis, such as PCA, and also to visualize the data, but they are not available yet. And finally, users can also select environmental data from all the environmental layers available in CartograPlant. So again, the environmental layers are classified depending on their location. Some of them are from North America and others are worldwide. So by clicking here, users can display all the environmental layers available, but we have more than 900 layers available. So in order to help you decide the environmental layers of interest, we have another new feature in CartograPlant that allows users to type directly here the keywords of the environmental data of interest, so that you can quickly identify the environmental layers of interest. Once you have decided what environmental layers you want to use for your data analysis, you can click on them, select them, and CartograPlant automatically generates a scatter plot showing the correlation between these variables. And this is very useful for data analysis in order to avoid this multicollinearity. So in order to avoid redundancies in your data, because sometimes two environmental layers or two environmental data are telling essentially the same story, such as, for instance, the temperature or the precipitation. And this way, that can help users to decide the environmental data to use in their data analysis. And finally, the last part is the data analysis itself. We have currently two analytical workflows available. It is possible thanks to Galaxy, of course, to a Galaxy instance, which is a very useful, very user-friendly platform to perform high-throughput data analysis without the need of coding skills. This is why it is very user-friendly. And we have essentially two analytical workflows. One of them is the genome-wide association analysis, and to do so, that is the association between the genotype and the phenotypes. And we make use, CartograPlant makes use of a program called Emacs that enables to correct by population structure and also by kinship, which is very convenient for a wide diversity of studies, of study approaches, and also the multiple testing correction, which it is also key for this kind of association analysis, because sometimes, since we are using p-values, sometimes if we are analyzing a lot of SNP data, just by chance we can we can encounter a high amount of false positives. So this is why to correct by multiple testing. So I'm going to show you how to perform this genome-wide association analysis with this Emacs program in CartograPlant. So once you select the workflow of interest, you can see that there is a brief description of what this workflow does, a brief explanation, and also the input files that are necessary for this analysis. So in this case, a genotyping file containing the SNPs in BCF format. Also, the SNP files filter by linkage disequilibrium in order to calculate the kinship matrix, because again, Emacs corrects by kinship, not only by population structure. And also, of course, a phenotypic file, because we are going to associate the genotypes with the phenotypes, and the population structure that we have already calculated. So once you have selected all your files available in your workspace, you can click Initiate Analysis Job. And again, all the output results are available here and also in your workspace in the Manage tab, so that you can just download them in your computer by clicking on them. And also the status of the analysis is displayed with a color code. So the files that are not ready are in yellow, and the files that are completed are in green. And again, all the output files are available in your workspace for download them in your computer. And that's it. Well, thank you so much for your attention. I would like also to thank all the team members of the project that have made this project possible, and of course to you all for your attention. And if you have any questions, please do not hesitate to let us know. Thank you so much.
Thank you, Irene. So Irene is our last presenter for our workshop today. And so we definitely want to open it up to any questions that you guys have at this time. Please, you can share them in this chat, or you can, I think, shout them out to us if you'd like to do that as well. So I'll just give you a few minutes to do that. And otherwise, I want to thank you for joining us today. You can also reach out to us via email, go visit some of the links that we provided attached to the workshop in order to get more information. And I believe also all of the recordings will be available for this workshop, whether or not you are registered. Irene, did you want to take any of the questions in the chat?
Okay, yeah, I'm just checking them.
I think we have just, well, maybe.
Oh, just one. Okay, is it possible to test multiple statistical models for the regression? For the genome-wide genome to phenotype regression model, I suppose no, because we only have a Emacs program, so there are not more approaches. We can include them, so you can suggest them if you want to ask, like, include this approach because I'm interested in, but so far we only have this one, but it is possible to update CartograPlant, of course.
Yeah, one of the nice things about using the Galaxy framework is that as long as we have, if there's an open source package that you're interested in adding to our workflow, let us know. It's not quite as simple as just installing it, of course. The way that we build the workflows is that we're funneling data, in at each step through that process, and also making sure that you're not presented with options that are not relevant to the data sets that are in for analysis at that time. But if there's other models, and there are many options in this space that you would like to see available there, let us know, because that's something that we can start to develop into the workflows. Any other questions? All right. Maybe at this time I pass it back over to Nicole with anything else, but I think we've covered everything on our side.
Yeah, thanks everyone for a really great workshop, and thanks for some great questions in the chat as well. So I'll leave it open for another minute or two if there are any questions. As Jill said, feel free to type a question into chat, or just go ahead and unmute yourself and ask a question if you are comfortable. I will also note that at one point, I believe it was Jill put in an email into the chat, so that if you did have any questions regarding CartograPlant, then you could kind of reach out to them that way as well. Maybe stewing over the recording or just thinking about everything you've heard over the last couple of hours, we'll jog something for you.
Yeah, absolutely. And I just put that email there. And also, you know, a lot of the things that I think people have mentioned or directly messaged me about is, you know, can I bring in this layer or that layer? So tell us what kind of layers you're interested in that you're not seeing there. We want to get those in. And analytic tools, and just related to the question that we just had. So those are the two areas that I know that will have, you know, users interested in different aspects as well. So definitely send those and any other questions that you have as well. But one of the big things is we would definitely like to get your, you know, getting your data into the system, submitting your studies in through this FAIR workflow will make them available to you to perform meta and mega analysis against other studies. So if you're having trouble working with trying to get a study in, if you'd like to get a study in, you don't know where to start, we'd also love to hear from you on that as well. And I want to re-emphasize that all of the studies that gets submitted to us get a DOI. We process these things through Zenodo. So you have a DOI associated with the entire study and the metadata package that you can refer to in your publication. So that will go on in there and live on and be available even outside of TreeGenes. So definitely make use of that resource for your own FAIR data reporting as well.
Great. I love to see you guys making things super easy for everyone. It's great.
We're trying. Let us know if it's not, but we are trying. So we're happy to get feedback.
Well, I'm not seeing questions. I'm not seeing anyone unmute. So I think we'll just go ahead and end it here. Everyone wins back 14 minutes today. And yeah, again, thanks for a really great workshop. Thanks for attending, everyone. And as I've said before, a recording of this will be made available later today, whether or not you're registered for this. So tell your friends, I guess.
Perfect. Thank you so much.
Everyone.
Thanks, everyone.
Thank you.
Thanks, guys.